Welcome everyone to this 2024 seasonal update webinar for the Southern Region presented by the Australian Fungicide Resistance Extension Network, also known as AFREN. I'm Bridget Penner from Ag Communicators. In today's session, we're pleased to have some leading experts in cereal and pulse diseases in the Southern Region here to present. So we'll be joined by Dr. Tara Garrard, who leads the SARDI cereal pathology team, Dr. Harry Dadu, who is a senior research scientist in the field crop pathology team at AgVic in Horsham, and Dr. Josh Fanning, who is the plant pathology research leader at AgVic. So for those who aren't aware, AFREN is a GRDC supported initiative that provides up-to-date resources and information directly to Australian growers, agronomists and advisors. You can find a whole heap of resources, workshops, the Fungicide Resistance Management Guide, webinars, fact sheets, podcasts at afren.com.au. So before we kick off, I just wanted to direct your attention to the Q&A box in the bottom of your screen. So if you have a question, please feel free to pop that into the Q&A box and we will look to cover those at the end of the session. So without any further ado, I will hand over to you, Harry. Thanks.
Thank you, Bridget. Good afternoon, all. I'm Harry Dadu. Today I will be talking about cereal diseases, mostly focusing on the factors that drive the disease for the rest of the season. And also I'll focus on a little bit on rust management and septoria and net blotch management in barley. To start with, what are the factors that will drive disease impacts for the rest of the season? In my opinion, pathogen is about referring to the inoculum levels that are present in the paddock, and climatic conditions, most importantly rain, and how humid it would be for the rest of the season, and varietal resistance, whether it is a variety that is resistant in your paddock or a susceptible variety that you're growing. And the other factor that I base, since it is a season that was dry, I'll put my money on yield potential as well. That was for that paddock that you're in. I'll discuss more on pathogen as I go through in my presentation, but I'll talk about climate and host plant resistance and yield potential in coming slides. So looking at climatic conditions for the southern region, not very great news of what we had in terms of winter rainfall. It was pretty much dry for most of the Victorian and South Australian region. As you can see from the graph, there's a lot of red places which refer to very much below average rainfall. So which means we had late dry sowing mostly, and also late emergence. And because of late emergence, there was poor crop establishment as well. And that also meant the conditions were not really conducive for the disease. So which means the disease was, there was little delay in the disease onset in these regions. But however, because of on the back of last 3 seasons, which were wet and which were very conducive for the disease, we still have a lot of inoculum, which we are still dealing with. And this is going to be a little important in the season too. And what's the season looking like for the rest of the spring? Not really sure if many of you had a chance to look at the latest release from Australian Bureau of Meteorology. It shows it was released on Thursday. So they say there's nearly 60 to 75% of chance of exceeding the median rainfall for spring season, so which is quite good news. I hope it stays that way. But there was at the start of the season, it was said it will be wet spring. Then it was changed to dry spring again. And then now it says sort of as wet spring for the southern region. So if that all happens, it'll be really nice. So what rainfall actually means for disease? So rainfall does bring a lot of humidity to the crop canopies, which actually is helpful for the pathogen to grow. And also it brings leaf wetness, which is also important for the germination of the pathogen. And then rain itself will help to spread the disease to the other crops. So that is how rain will be very critical. And spring rainfall will be very critical for one of the main reasons is as the crops move into stem elongation and reproductive phase, where the crops put all the flag leaves out. And those flag leaves, if you have disease, any of the disease on those flag leaves will be very important because these flag leaves will be contributing to the yield. So it is recommended that we protect these flag leaves or these critical growth stages at this point of time. And should the spring turned to be very wet. And the other factor that I mentioned about was host plant resistance and yield potential. As pathologists, we always bat for using resistant varieties in your crops. The reason is because they reduce risk of yield loss, as shown in the graph that was presented from spot form. So it shows that if you're using a highly rated resistant variety, you are not going to get any yield loss because of the resistant variety. Whereas if you're using a highly susceptible variety, you're bound to get nearly 20% of yield loss, irrespective of the situation. So it is very important that we need to use resistant varieties in the crops. It reduces the risk of yield loss, one thing, and also it reduces the usage of more fungicides. And at the end of the season, it also builds up less inoculum levels going into the next season. So these are the benefits that we get from using a highly resistant rated varieties. Having said that, the other factor that I want to mention is yield potential. So given that this season has reduced yield potential across the southern region, you might need to plan for low yield potential. If you have low yield potential, what we have seen generally is disease doesn't really bring that much penalty to the yield to your crop. So this example, particularly from spot form, this is the data that is generated from nearly 10 years worth data, also coming from across Australia. So where it shows that if you have a paddock which is less than 3 tons per hectare, you won't be seeing any yield loss from regardless of how much disease you have or regardless of the varietal resistance that you generally have in your crop. So we know this season has been very dry so far and the yield potentials have reduced. So probably when you're making your fungicide decisions, always keep in mind what is your yield potential. So if it is less than 3 tons for most diseases, it is useful that you don't need to spray a fungicide for controlling disease. So those are the important factors that what we have seen or what will be driving the disease and disease impacts for the rest of the spring season. Coming to the disease that we have seen or observed in southern region for the 2024, the first important disease that what we are seeing currently is stripe rust. Stripe rust had relatively late detection compared to what we have seen in 2022 and 23, but still it was relatively very early compared to the long-term average of last 20 years. So it was detected in June in Eastern Australia. And now where we are in August, it has spread all across the Eastern Australian belt. As the map that was shown on the right side, it is coming from University of Sydney interactive map that they put out every 2 weeks. It doesn't show any spots or any locations being tagged for South Australia, but as Tara said this morning, it has been found in all the growing regions across South Australia. So the map might get updated. And also the map shows that it is not just this stripe rust. There are a few leaf rust, and also a few barley leaf rust which have been detected in New South Wales. So depending on how the season will end, if it is a dry and warm, then we might expect more of leaf rust to be seen in southern region as well. And the same goes with barley leaf rust. Talking about the management strategies for cereal rust, I think it's very important to monitor the crops for symptoms. I have found some symptoms this morning in one of the crops that I have surveyed. So once you know that you have seen the symptoms, know the varietal resistance, whether it is highly resistant or susceptible variety. Based on that, you make your fungicide strategy and also understand what are the conditions that are available at that point of time. So if you're growing a susceptible variety, definitely you might need to consider a spray if the crop is still at stem elongation and growth stage. And also the detections have been coincided with mostly where the susceptible varieties are grown, for say example, Scepter or Rockstar or Vixen. And also it is identified with those paddocks were not being sprayed with an AFREN fungicide, which is a flutrifol. So if you have noticed those symptoms, probably you need to take some care for those paddocks. And there is another decision support tool which we generally recommend for people to use. That is stripe rust weed management app, which was developed by GIDC and DEPAD Western Australia in collaboration with Agriculture Victoria as well. So where we have provided decade-long data to go into that app. So the app actually helps you to make a decision on whether to spray a fungicide or not to spray a fungicide. So it takes into consideration about the paddock conditions, like varietal resistance, what is the disease, and also the weather forecast. So it tells you, by inputting all those details, it tells you whether you have to spray for the disease or not. For example, if the crop is at stem elongation growth stage and you have seen the disease and it's a susceptible variety, it allows you to go for a spray and also provides a probability of the profit. For example, if you're doing a spray or if you found the disease late in the season, like at flowering stage, you probably don't need to spray. So that's what the app will say essentially. So yeah, use it. There's a lot of feedback from a lot of growers in Victoria in the last 2 seasons, and it might be helpful in a season like this. Talking about the other disease that we have seen in Victoria and South Australia, it is the next important disease that we are seeing is septorated red blotch and powdery mildew. Tara will be speaking on powdery mildew, so I'll focus on septorated red blotch in my presentation. Septorated red blotch has been a disease very common in high rainfall regions. What we have seen in before 2020, it was a really big disease in those regions and also continues to be now. But it was not a disease which was common in medium and low rainfall regions in Victoria and South Australia. But what we have seen since the last 3 seasons, there is increased prevalence or incidence of septoria. One of the reasons was use of susceptible varieties, and the other thing is the conditions that have been really conducive in the last 3 years because they have been very wet, and because of which we have seen a lot of inoculum buildup in the regions, and also there was more disease impact in the crops that are being grown. And this is a graph which shows that yellow leaf spot was a disease pre-2020s. But what we have seen in the last 4 years, yellow leaf spot has been seen significantly reduced. This is because, once again, the use of varieties that we are using at the moment, the way we are using better resistant varieties for yellow leaf spot compared to septoria. Now moving on to the disease impact of septorated red blotch in terms of yield. So we have done, in collaboration with the South Australian Department and also GIDC, we have done 3 years of research work in field at the moment, where we have done tested different varietal resistance and also tested against high disease and low disease environment. So what we found is the varieties that are susceptible are going to lose some yield because of the disease. And this data is coming from 3 years worth of seasons. In the first season, which is 2021, we didn't see much of yield loss in Lancer, which was highly resistant rated variety for septoria, whereas for Hammersail Plus, we didn't find, even though it was moderately susceptible. But the yield loss was found in Scepter and also Impala, which were probably susceptible varieties at that time. But as 2022, everyone remembers it as a wet season. And also, it was also very conducive for septoria. And what we found was nearly 35% of yield loss in Scepter and also Impala, which are highly susceptible varieties. And the other thing, we also found some yield losses in Lancer and Hammersail Plus as well. So it shows that it is, as I said earlier, it is a high rainfall region disease, but now it has moved to the Wimmera or medium and low rainfall regions, where we are getting seasons which are above average rainfall. So in seasons where rainfall is above average, you might need to consider controlling septoria in your paddocks, and especially if you have susceptible varieties. And also going back to the yield potential thing that I said earlier, so we did find some really good data from South Australia where they had yield potential of less than 3 tons per hectare, and irrespective of the varietal resistance and also irrespective of the amount of disease that was present in that variety, we didn't find any yield loss if the yield potential was less than 3 tons per hectare. So that shows that if you have disease in your variety and also the yield potential is very less, then you probably don't need to worry about septoria, and also probably don't need to put a fungicide to control it. And if the yield potential is more than 3 tons per hectare, then probably I would say, depending on the variety and the conditions that are prevailing, I would probably suggest a fungicide. Now, talking about fungicides, we did test what fungicides probably we need to use at what growth stages. So in terms of septoria control, we have found the benefits will be very good when the seasons are really conducive for the disease. For example, 2022, where we found the highest yield gain, if we are using a dual foliar spray, one at stem elongation and the other at flag leaf emergence. So we did find nearly 37% of yield gain in that season in Victoria. In 2023, we still had better conditions in winter, which was high above average rainfall, and we did find nearly 14% of yield gain. We also tested these scenarios in Mali, which was a low rainfall region, where we didn't find any yield loss in 2021 nor in 2022. And probably we did get a really good season in 2023, and we started to see some yield benefits by using fungicides. That shows the extent of disease is spread across Victoria at the moment. So it is now detected in. It is also present very well in southern Mali, or established very well in southern Mali. So probably those growers who are in low rainfall zone, they also need to think about septoria if the conditions are really there for the disease, which was not the case in the past. Talking about fungicides, there is a factor that fungicide resistance that we are experiencing in the recent times for septoria triticide blotch. So in the past, we have found resistance to flutriafol, tebuconazole, and propiconazole for septoria in the southern region. And what we have seen in the last 3 years, there is increased reduced sensitivity for epoxiconazole, which is routinely used for controlling septoria. So there is a chance that if you're using epoxiconazole, you might see reduced performance. And if you detect that, this is because we have detected a reduced sensitive mutation for septoria. And it is spread across South Australia, southern region, actually. And yeah, it is suggested that people do consider fungicide resistant management strategies when applying or when controlling for septoria. Also, there is a news that quinols have been found to be reduced sensitive resistance to quinols has been found in southern Australia, and also this has been now detected in Tasmania, which means all the strobilurins that you use might be showing reduced performance. But good news is it is not yet detected in Victoria. So it is not detected in Victoria means we can still freely use, but probably we can't use more than once in a season. So make sure that you use these fungicides only one time in the season, either even if you're using as a seed treatment, so that you get better control or you reduce the fungicide resistance development. That's about septoria triticide blotch. Now I'll quickly talk about barley diseases. The diseases that we are seeing in southern region at the moment: net form net blotch, spot form net blotch, scald, and leaf rust, probably upcoming. I should not say that it has been seen yet. And then what we have seen is loose mud. Many growers on the Twitter have been updating that they are watching loose mud in most of the crops. And rhizoctone is a root disease that we have found some samples coming to us from southern region. So those are the diseases, but most importantly, the diseases that are topping charts are net form and spot form at the moment in southern region. So I'll talk about net form, net blotch, IDM, and also fungicide resistant status. In the last few seasons, again, very similar to septoria triticide blotch, what we have found is there is increased prevalence and incidence of net form net blotch in the southern region. So in the past, we have seen nearly 10% of crops infected for net form in 2019 for say, but what we have seen since then is increase in prevalence. This amounts for nearly 51% of crops surveyed in 2023 were found with net form net blotch. And also the severity has increased from 5% to 27% at the moment. So this shows that there is real potential that net form can become a really big problem. And one of the reasons is because the amount of area that we have under susceptible varieties, for example, RGD Planet. And the other thing is the conditions that are there for the disease to spread. So this disease, once again, like septoria, likes wet conditions. So if you have a season which is really wet, then we probably expect more yield losses from net form. And In terms of impact for the yield, so we do have some results from last year where we have tested RGD Planet and also compared it with better rated variety, that is Titan AX. So what did we find? So we found nearly 22% of yield loss when it is not treated with a fungicide, whereas Titan AX, we didn't find any yield benefit by using a fungicide. So regardless of the disease that was present on the crop, there was zero yield loss. So this once again reiterates that message that avoid highly susceptible varieties if you want to get less risk of disease and also less risk of yield penalties. This also goes the same for spot form of net blotch. If you're using a highly resistant variety, we probably don't need to worry about fungicides. And talking about the fungicide timing, so we did have really good data on fungicide timing. So in 2000, this was the data from 2020. So what we found was using a Systiva treatment as a seed treatment was found to be best for controlling net forms or spot form as well. And also a follow-up spray with if you're including foliar sprays. So we found benefit of using foliar sprays at 31 and 39, as we found with Septoria, rather than using anything, rather than not using any fungicide. We did find really good benefit by using these strategies. But what is happening in 2023? So as I said earlier, we started to find some fungicide resistance. So we found some really good, no, I should not say really good, some resistant pathotypes for net form. And what we found in the trials that we conducted in 2023, Systiva was found to be unresponsive for controlling the disease. And hence we found nearly 16% of yield loss because of in those treatments where we had Systiva only. And that means there is more stress on foliar applications. Of course, still the foliar application does work, but we still found some reduced sensitivity or resistance for a couple of DMIs that includes tibuconazole, propiconazole, and even prothioconazole. So this means that going forward, fungicides might be less reliable, especially group 7 and group 3s or DMIs or SDHIs. So probably take into consideration what is happening in your region and talk to your pathologist or send samples to us. We'll be able to detect the fungicide resistance if it is available in your paddock and probably go from there. But once again, the message is always use highly resistant rated varieties. This means you won't need to worry about using a fungicide for controlling these diseases. And as I said, we found Systiva resistant and also even DMI resistant pathotypes. So this is the data that is coming from 2023 across Victoria. So what the current status shows that the sensitivity is nearly only 54% populations are showing sensitive, whereas for SDHI, it is only 41% of populations are showing sensitivity towards SDHIs. This means that these fungicides can still work, but they should be managed really well. If you're using overdose or overusage might cause these levels to increase to 100%. So probably once again follow the fungicide resistant management strategies to reduce the disease. At the moment, if you look at the map of Victoria, the locations where we have detected fungicide resistance, so those red dots in the map show that those are the regions where we have found samples which are showing some resistance to both Systiva and also even for group 3, which is DMI fungicides. So this shows that it is quite widespread across Victoria. So yeah, make sure you follow that fungicide resistant management strategies. The other bad news is BASF also detected some resistance or reduced sensitivity for group 11 fungicides in South Australia. So that was for net form. So this means that in future, we might lose the activity or group 11 fungicide activity towards net form as well. So make sure that it is not going into that level by reducing the usage. And this also means that the other regions where it is not detected, like Victoria, for say, we need to follow that fungicide resistant management strategies so that we control the disease well, and also we don't get this fungicide resistance into our region. With that, I'll pass the baton to Tara to continue on net blotches and powdery mildew.
Thanks very much, Harry. It's great. So I'll start off by just touching on net form, net blotch, reduced sensitivity and fungicide resistance in SA, and then talk about barley leaf rust and wheat powdery mildew. So similar to in Victoria, South Australia has detections of the SDHI, reduced sensitivity and resistance right across the state, and getting reports from the beginning of this season where Sestiva was applied as a seed treatment, that net form net blotch was still starting very early in some of those crops, particularly in the Mallee region, suggesting that we're seeing the resistance there as well. So as far as we know, it is spread throughout the state. Be aware of it and kind of monitor your paddocks. If you are using Sestiva, then make sure you've got plans to use follow-up applications of other modes of action as well. Barley leaf rust, this is pretty common in the southern region, particularly later in the growing season as temperatures start to warm up. We know that we have the sexual stage of the pathogen in South Australia, and I think Harry's mentioned in Victoria as well. This is the Star of Bethlehem, which is this funny little weedy plant that you can see here on the screen. Essentially, this hosts it over summer, meaning that we don't need the same system with volunteers that we do for, say, wheat stripe rust in order for the pathogen to continue and get started each season. It also allows for recombination of the genetics in the pathogen and increases the diversity of pathotypes that we have. So we know that we've got quite a lot of barley leaf rust pathotypes throughout the southern region. The University of Sydney have done some work in the last few years at the cereal rust program there, looking at reduced sensitivity to DMIs in barley leaf rust, and have found that we've got insensitive pathotypes tracing back to a particular lineage in Western Australia since 2001. And as they've looked further at the barley leaf rust samples across Australia over the years, they've found that it's actually prevalent right across the barley growing regions and shows increased insensitivity. It displays resistance to all 8 of the DMI actives at field rate and high field rate concentrations. But combinations like Amistar Extra, Aviator X Pro, where we've got a different mode of action there, are still effective, and we know that those other modes of action aren't affected. So we're saying use some caution with your applications of barley leaf rust. But in saying this, I think whilst it's new information for us to know that we have these reduced sensitivities present in barley leaf rust, it's probably not new to what's been happening in our system. I mean, these detections date back to 2001. So realistically, we've been adapting and working to these reduced sensitivities in our management systems and our fungicide applications over the last 20 years. So keep doing what you're doing and keep an eye on any changes in the sensitivities of your DMIs for barley leaf rust. So yeah, it's good for us to know, and a bit of a word of warning, and keep an eye out, but it's not necessarily a new thing. It's just that we have more information about it now. So having a look at the status of fungicide resistance across Australia and in the southern region, we're now going to move on to wheat powdery mildew and talk a little bit about what's happening there. So we know that we've got reduced sensitivity and resistances in the southern region for group 11 and group 3s. And we'll also touch on some of the management strategies that we've got available to us given these fungicide resistances. So the strobilurin resistance is certainly one of the ones that's the most talked about, and the strobilurins have been quite commonly used for control of wheat powdery mildew in the last 5 years. What this map shows, which is a combination of surveillance work done by Agriculture Victoria, New South Wales DPI, Trengo Consulting, and fungicide resistance testing work by the CCDM have mapped the prevalence and the frequency of the mutation that causes this resistance across the southern region. And on this map where we have red spots, that's showing that 0% of the wheat powdery mildew pathogen population contains the mutation for resistance, meaning that it's not present in the pathogen population. As we move up to blue, we move up to 100% of the pathogen population in the sample tested contains the mutation. So as we move from 0 to 100, then we're getting less and less sensitivity to the strobilurins when we're using them. So what we can see here is where we've got orange, green, yellow, and blue. We do have that present in the pathogen population, and we're likely to get reduced activity from strobilurins. It's a little bit hard to see on the scale of a computer, but we do have on the Eyre Peninsula some yellow, green, and oranges through here. The same with the Yorke Peninsula and the Upper Yorke Peninsula. You can see more of that yellow and green popping up in there, and we know that northern Yorke Peninsula area has kind of been a good study spot for the emergence of these mutations. Moving then into the Mallee and upper southeast, we also have pretty common 20 to 40% of the pathogen population. And moving into Victoria, we can actually see no reds, and that all the samples that were submitted there did actually have this mutation present in the pathogen population, much more severe down in the southern parts of Victoria and the high rainfall zones. So be aware of that, and the way that we kind of work with that is understanding that it's likely going to be present. What percentage of the pathogen population you actually have with the mutation in your paddock is going to make a difference then to how well it responds. So working to keep those levels down as low as possible with the selection pressures that we put on the pathogen will help to give us more longevity of this fungicide. So combining our modes of action is key here, and trying to not use susceptible varieties where possible. We do know as well that with the DMI reduced sensitivity, that is prevalent throughout SA, and it's got a pretty high prevalence level. Now, the difference here is that we're working with multiple mutations for the DMIs that take us from sensitive through to fungicide resistance. And so a lot of this testing is on indicator mutations that give us an idea about some of the mutations that are there, but it's the accumulation of multiple that really brings us to the fungicide resistance level. So even though there's a high prevalence of it, we're going to have varying levels of the modes of action that are still containing sensitivity, and at different levels of that sensitivity as well. Having a look at propiconazole, the CCDM had a look at how a wild type isolate that did not contain the resistance mutation versus ones that did compared, and you can see that at 70 mils per hectare as well as 140 mils per hectare, there's still plenty of pustules that are developing on those wheat plants. So certainly that translates from the genetic detection of the mutation across to what we actually see in the field. How we actually work with this and manage that in paddock practice is where it gets a lot more tricky, because it's obviously quite technical, the mutations that are involved and where they are and at what levels spotted throughout our growing regions. So really we're looking at combining the modes of action as much as possible and not repeating modes of action over and over again, particularly within the same growing season. And Harry was talking before about how the levels of wheat yellow leaf spot have changed over a number of years that they've observed through their surveillance activities. I'd say we've definitely seen an increase in the amount of wheat powdery mildew across the last 5 years particularly. And with yellow leaf spot, we've seen much better wheat resistance genetics coming into rotation with our varieties, which has really helped to reduce those levels down. With powdery mildew, we're experiencing the opposite, where we've seen a lot more susceptibility in our commonly grown varieties over the last 5 years. And with that, we are seeing the levels slowly increase over time. We're at a point at the moment where we have quite high inoculum levels in most of our growing regions of wheat powdery mildew, and growers are really struggling to manage this pathogen. And when you have a look at some of our commonly grown varieties here, you can see the number of SVSs, so susceptible to very susceptible varieties. Now, if we go back to mace, when that was commonly grown quite a while ago, you can see that's actually an MSS. Now, there's a big difference when you've got a large portion of the area planted to MSS varieties versus SVS varieties. So we're really encouraging growers to now move away, if possible, from SVS varieties as we've got more options becoming available. Even if you can move up to an S or an MSS, then we're slowly going to shift the inoculum levels back down again. We do also have, under the emergency permits, 3 new actives available to us for controlling wheat powdery mildew. These are completely different chemistries to the DMIs, strobilurins, and the SDHIs that we previously have and currently have registered. These are specifically mildewicides, and so they shouldn't have an impact on other foliar pathogens. So keep that in mind if you are using these and applying them, because you will need to use something else to target other foliar pathogens if they're present as well. The price point on these, however, is a bit higher, and that's partly because they've come across from horticulture and other industries. But it's really important that we protect these chemistries. They are not immune from developing fungicide resistance as well. So please keep that in mind. Use them cautiously. Use only one application of each chemistry within a growing season, and making sure that we're following up with different modes of action as well. So they are there to help to control the issue while we've got a lot of fungicide resistance and susceptible varieties being grown. So despite all of that, how do we manage wheat powdery mildew? Variety selection, this is kind of the #1 thing. We really need to look at what are the highest pressures in our growing system, and can we move away from SVS varieties for those high pressure diseases. Don't expect in a conducive season that you are going to get complete control of wheat powdery mildew if you're growing an S to VS variety. Even with fungicide use and really good spray strategies, you are still going to have some levels of the disease present throughout the season. Paddock selection, so a stubble-borne disease, so stubble retention systems are really crucial and can contribute to your disease risk, so manage your rotations. Prepare a fungicide strategy. The key with wheat powdery mildew is preventative and being really proactive. So rotating our modes of action and combining actives where possible, targeting our key timings. If we can hit before canopy closure and get down at the base of the canopy, we're going to hit those basal stems where powdery mildew hangs out. And the proactive approaches are much better here. We know that our chemistries have better protective effects than they do curative effects. Application types, so lower water rates have less penetration through the canopy, and with wheat powdery mildew that hangs out at the base of the stem, that can mean it can be harder to actually get the target down to where the pathogen is. So keep that in mind. Essentially, there's no silver bullet, and this is a case where fungicide resistance is really starting to challenge us in our growing system. And so bringing it back to some of the core principles of managing fungicide resistance, which is helping to prevent new occurrences of fungicide resistance, but also for the ones that we already have, providing selection pressures that keep this down as low as possible in the pathogen population and give us the most use out of the chemistries that we've already got. So to do this, we're looking at variety selection being the most important tool that we have available to us, and that should be the largest part of our decision-making process when it comes to controlling disease. Secondarily, we've got the other strategies that can be used, such as crop rotation, managing green bridges, stubble management, later sowing, whatever levers you've got to pull in your farming system that can help to reduce that disease pressure, you should be considering after your variety selection and before your fungicide program. Lastly, fungicides are a tool. We should only be applying them when necessary and applying them strategically. So we want to be rotating our modes of action, using mixtures if they're available, and staying within our label rates. So that's all on wheat powdery mildew. Just in terms of SARDI, if you've got any diagnostic queries, then you can get in touch with either our pulse and canola crew. Most and Carney's details are here, or with myself through the cereal pathology team. We're also collecting disease samples. So if you'd like to send some disease samples through, then some of the details are there. They're also on our Crop Watch page through the PIRSA website. And thank you to all the contributors to this research. I'm now going to hand over to Josh Fanning. Thank you.
Thanks, Tara. So I'm going to be talking about pulse disease management, but I'm going to be giving a really quick presentation today because on Friday I'll be giving another presentation, which will be in a lot more detail across Australia with Levente from University of Southern Queensland. So I'm just going to focus on what's an issue at the moment, or what could be an issue in the southern region. In terms of fungicide resistance in pulses, we're very lucky in pulses. We don't have fungicide resistance that's causing field failure at the moment. We've seen some lab detections, but essentially it's not really an issue in the field. We definitely do see reduced efficacy of fungicides in the field, but every time we've tested those, it's usually due to really conducive environmental conditions allowing for disease to progress. So firstly, I'd just like to go through though, is it really disease that you're looking at? Correct identification of disease is probably the most important thing. And I've put a picture up there at the moment of basically physiological spotting in barley. I don't know where the picture's gone of physiological spotting in chickpeas, but what are we actually looking at when we are looking in the field? Are we looking at viruses? Are we looking at abiotic stresses, water stress, nutrition, and herbicide? And given the season that we're having in the southern region at the moment, we've got some areas like in Victoria, or some areas of Victoria that are on good soil moisture. We're not exhibiting that much early at the moment, but in South Australia, inquiries are already coming through of that moisture stress and conditions caused by that moisture stress, or what we would expect. And the other thing that we can often see is exacerbated symptoms of herbicide damage when we're not getting that rainfall in season, such as seasons like this year. So it's ensuring that when we're actually identifying what we're seeing in crops, it is disease before we're actually undertaking our disease management. The other thing that we're starting to see is soil-borne diseases come through. I've just put up a few of the pathogens that are causing soil-borne disease at the moment. And given the season that we are having, when we're not being able to access or we're not getting as much in-season rainfall in certain areas, then our soil or our root structure is actually having a fairly important part in that moisture capture and that moisture uptake into the plants. So our soil-borne diseases are actually going to result in symptoms above the ground or in other stresses, such as moisture stresses, or some of those other physiological or abiotic symptoms that we may see on plants. So it's important to dig up your roots and have a look and just see and make sure what you are actually expecting to be disease is disease before treating it. So in terms of diseases in pulses, our biggest issues are probably ascochyta and botrytis. So we've got some pictures of ascochyta blight there on the screen in faba beans, chickpeas and lentils. It's usually an earlier in the season disease, but can progress throughout the season, controlled with varietal management, similar to our integrated disease management, ensuring we're sowing clean seed. But essentially, they're the symptoms there. So it's basically a white necrotic lesion with black pycnidia in it. And we can see those pycnidia even on seed defects. We can see them on the pods or the leaves like pictured there. Or botrytis grey mold, and here's some pictures in lentil. Essentially, we're seeing a mouldy crop. We're seeing what I would call as a pathologist, lovely grey fluffy mould, but to a grower, that's certainly not lovely to see in your crop. Or we'll be seeing those white lesions on the leaf tissues, and those white lesions won't have pycnidia. So very different to our ascochyta symptoms. In chocolate spot, which is also botrytis, slide changes, looks very similar as well to our lentils, except you don't see often that white fluffy mold as much. You'll see these chocolate lesions on the leaves that'll spread. We see a lot of spots on beans or faba beans, but how do we know that it's actually chocolate spot? We usually see sort of an orange to that chocolatey color of the lesion. We won't see the pycnidia, but when we really know it's chocolate spot, you'll see those brown spots appear on the flowers like you can see there in the picture. And that's how we really know that it's botrytis. And it can have significant impacts on seed quality. Both these diseases require lots of rainfall, and chocolate spot and botrytis in particular requires a nice moist or humid canopy, essentially. So we're not really seeing large amounts of these diseases across our regions at the moment, but if you actually go out there and have a good look, we are actually seeing the early symptoms of a lot of these diseases. And in Victoria, we saw a lot of ascochyta early in the season in some areas in high-risk scenarios when people growing lentil rotations or lentil rotations. And they require that we're seeing stem breakages, and they did require fungicide management even in our MRMS rated varieties. But as we progress through the season, in the last week in Horsham, we've certainly had skippy showers, or basically showers every day for the last week. So we're seeing disease progress a lot more. But in other areas, such as the Northern Mallee, we've been quite a while without a rainfall event. So our environment's playing quite a significant factor, and those areas aren't going to require fungicides, at least in our MRMS varieties. And until we get rainfall, our susceptible varieties will be pretty good for disease, and we won't be seeing much disease on them until we see those conducive weather conditions. The other thing that I like to point out with botrytis grey mold, ascochyta blight essentially is a separate species of ascochyta in each crop. But with botrytis grey mold, botrytis cinerea goes across most of our crops, or all our crops. So if you're seeing your symptoms exhibited of botrytis in your faba bean crop and you've got a vetch crop next door, or vice versa, you are going to see that pathogen come from one crop to the other, unlike ascochyta. That sort of just heightens our risk. So in our environment, if you've got a lot of susceptible varieties across your crop types in pulses, then that increases the amount of pathogen in the landscape, and that pathogen is going to more likely result in disease if we get conducive conditions. And botrytis moves very, very quickly, and that's why I've sort of focused on that. So being a quick presentation, how do we manage those diseases? Yes, we're not sowing clean seed right at the moment, but it's part of the integrated disease management strategy. So we've got crops in the ground next year for this year. And if we're looking at keeping seed for next year, we need to think about protecting our seed so we can sow clean seed next year. We don't want to be sowing disease back into the ground straight away. Thinking about those crop rotations and how much disease or susceptible varieties we've got in the landscape, then choosing the resistant varieties that we can. We're getting luckier in pulses. There's a lot more resistant varieties coming through the breeding programs, and a good level of resistance out in the environment, out in those released varieties that we can utilise for disease management. We need to be monitoring for disease, and that's Obviously more important in our susceptible varieties, but it's still important in our resistant varieties as well. And we need to be assessing our disease management strategies to ensure we are getting the level of disease control we require to reduce grain yield losses. So, and then it's understand our seasonal risks. So I talked about our medium rainfall in Horsham, where we've been getting those rainfall events, whereas in other areas, we certainly haven't been seeing that rainfall event, and there's certainly a lot drier areas this year. It's then about optimizing our fungicide strategy to those above risk factors, and then aiming to spray before our rainfall events, keeping in mind that plant growth post-spraying is usually unprotected. They're a preventative approach, not curative. And again, protecting that seed, both for a grain quality aspect, but also if you're keeping the seed for next year, so you can sow that clean seed. Varietal resistance, the best place to get the varietal resistance ratings is the NVT website or the Pulse Disease Guide from Agriculture Victoria. So I've put a QR code there, and they're released annually with the up-to-date ratings based on all the disease screening that happens nationally. So pulse disease management in fungicides, it really depends on those multiple risk factors that we've talked about before. Canopy closure fungicide is really our most effective fungicide application for both ascochyta and botrytis, but botrytis in particular, and then follow up if the risk requires. We get in those areas that are getting nice, or not so nice, I suppose, but warm, dry conditions, we don't really need to think about our fungicide. Essentially, that non-conducive environmental conditions is our best form of fungicide. There are a whole range of fungicide options. I'm not going to lay them out on the table here. We've put a field crop diseases article out, and I put it in the GRDC updates this year. This link here takes you to those fungicide options. And then essentially it's just remembering it is about that holistic integrated disease management for both our fungicide resistance. Yes, we're not seeing fungicide resistance commonly in pulses at the moment, but we need to prevent presentations like what we've just seen from Tara and Harry. And we don't need those presentations if we prevent that fungicide resistance right from the start. So I don't want to be standing there like them in a few years' time talking about how much fungicide resistance we've got in pulses. So if we start with our varietal selection, moving on to our non-chemical options, and then our fungicides as our last resort where required. So it's about thinking about that all through the season. And I'm not going to go over this in great detail as it's already been gone over. So with that, I'll hand it back to Bridget to move into discussion.
Thanks, Josh. Thank you for that. And thank you very much to Tara and Harry as well for your presentations. I think we'll probably wrap it up there. There haven't been a huge amount of questions come in during today's webinar, but that's all good. I think you did a great job in covering off current disease risks and fungicide resistance. So I think probably any questions have been answered, which is great. So I just want to draw everyone's attention to the chat box. So I've just posted a survey link in there. And as Josh mentioned, there is a dedicated pulse and mung bean webinar on Friday. So I've put the link in there too in case you're interested in registering. So on behalf of the AFREN team, I would like to thank each of you for joining us today, and thank you again to Tara, Harry and Josh for your presentations today. We look forward to seeing you again at another AFREN event in the very near future. Thanks everyone.